The immune system. Further information, look at immune system and immunology individual articles. So the immune system is a network of biological processes that detects and responds to a wide variety of pathogens. Many species have two major subsystems of the immune system. The innate immune system provides a pre-configured response to broad groups of situations and stimuli, and the adaptive immune system provides a tailored response to each stimulus by learning to recognize molecules it has previously encountered. Both use molecules and cells to perform their functions. Nearly all organisms have some kind of immune system. Bacteria have a rudimentary or basic immune system in the form of enzymes that protect against virus infections. Other basic immune mechanisms involved in ancient plants and animals and remain in their modern descendants. And these mechanisms include phagocytosis. See the pronunciation here?
Phagocytosis.
Yes, so phagocytosis, antimicrobial peptides called defensins. See the pronunciation here?
Defensin.
The defensins and the complement system. And let's take a note here. So, just note phagocytosis from ancient Greek phagogen. See this?
Phagine
or phagonin, to eat, and chitos, cell, is a process by which a cell uses its plasma membrane to engulf a particle, which is greater than 0.5 micrometers, or which is a millionth of a meter, giving rise to an internal compartment called the phagosome. See this here?
Phagosome.
It is one type of endocytosis, which is a process of bringing substances into the cell. A cell that performs phagocytosis is called a phagocyte. So here's phagocytosis, and this just uses the cell membrane to engulf a large particle. But in general, just anything that brings it inside the cell is just called endocytosis. So phagocytosis is a subset of endocytosis. So here's phagocytosis. You have a bunch of stuff. You have a star there. There's a large particle. There's a plasma membrane, and here's an overview of it. What ends up happening is the membrane engulfs this, and then it sucks it in, and now you have a vacuole that contains this large particle there. Very interesting. So antimicrobial peptides, that's over here. So these mechanisms of this immune systems evolved in ancient animals and plants and remain in their modern descendants. Yeah, so you have that phagocytosis. The other one's antimicrobial peptides, called defensins, and then the complement system. So this antimicrobial peptides, AMPs, are also called host defense peptides, HDPs, are part of the innate immune response found among all classes of life. They have been demonstrated to kill bacteria, enveloped viruses, fungi, and even transformed or cancerous cells. Very, very interesting. So these peptides kill these pathogens. And the complement system, also known as a complement, also known as complement cascade, is part of the immune system that enhances or complements the ability of antibodies and phagocytic. See this? Phagocytic cells to clear microbes and damaged cells from an organism, promote inflammation, and attack the pathogen's cell membrane. It is part of the innate immune system, which is not adaptable and does not change during an individual's lifetime. The complement system can, however, be recruited and brought into action by antibodies generated by the adaptive immune system. The complement system consists of a number of small proteins that are synthesized by the liver and circulate in the blood as inactive precursors. So when stimulated by one of several triggers, proteases, see pronunciation here? Proteases in the system cleave specific proteins, so these are enzymes there, to release cytokines and initiate an amplifying cascade of further cleavages. The end result of this complement activation or complement fixation cascade is stimulation of phagocytes to clear foreign and damaging materials, inflammation to attract additional phagocytes, and activation of the cell-killing membrane attack complex. About 50 proteins and protein fragments make up the complement system, including serum proteins and cell membrane receptors. They account for about 10% of the globulin fraction of blood serum. Yeah, which is this is quite a next level system right here of defense. So stimulated by one of several triggers, proteases in the system cleave proteins, and then it releases these cytokines and initiate amplifying cascade of further cleavages. So basically, this cleaves proteins, releases this, and then initiates even more of these to just cleave the proteins. And then you have a complement system of a whole bunch of proteins, and then the stimulation of phagocytes to clear, absorb, and then engulf the pathogens, etc., as well as inflammation to get even more phagocytes, and activation of this cell-killing membrane complex. All right, so you got a whole bunch of stuff happening at once. And they count for 10%. So these proteins, about 50 proteins and protein fragments make up this complement system. And they are 10% of the globulin fraction of blood serum. A protein precursor, note that a protein precursor, I mentioned here. So the complement system consists of a number of small proteins that are synthesized by the liver and circulate in the blood as inactive precursors over there. Put this bold there. So a protein precursor, also called a proprotein or propeptide, is an inactive protein or peptide that can be turned into an active form by post-translational modification, such as breaking off of a piece of the molecule or adding on another molecule. The name of the precursor for a protein is often prefixed by pro. So whenever you have the word pro, it's the name of the precursor for a protein is often prefixed by pro there. Yeah, which is interesting there. So pro-protein, and you add that in. So examples include proinsulin, precursor to insulin, what's the pronunciation here?
Propionmelanocortin.
Yeah, propionmelanocortin, which are both pro-hormones, so basically precursors to hormones. And so note here, protease, this right here, pronunciation.
Protease.
It's a protease. So a protease, also called a peptidase or proteinase, is an enzyme that catalyzes, which increases the reaction rate or speeds up, proteolysis.
Proteolysis.
Or proteolysis, which is breaking down proteins into smaller polypeptides, or single amino acids, and spurring the formation of new protein products. They do this by cleaving the peptide bonds within proteins by hydrolysis. Hydrolysis is a reaction where water breaks bonds. Proteases are involved in many biological functions, including digestion of ingested proteins, protein catabolism, see this? Catabolism, which is a breakdown of old proteins, and cell signaling. All right, and now this cytokines that was mentioned right over here. Yeah, so when stimulated by one of several triggers, proteases in a system cleave specific proteins to release cytokines and initiate an amplified cascade of further cleavages. So cytokines right here. Cytokines are a broad and loose category of small proteins important in cell signaling. Cytokines are peptides and cannot cross the lipid bilayer of cells to enter the cytoplasm. Cytokines have been shown to be involved in autocrine, paracrine, and endocrine signaling as immunomodulating agents. So autocrine, that's the within cells. Paracrine is close by. Yeah, and endocrine is within the circulating system. So as immunomodulating agents, their definite distinction from hormones is still part of ongoing research. So basically, hormones, but small proteins, but basically they're more broad and loose category of small proteins, and they're peptides and can't cross the lipid bilayer, etc. Immunomodulation is modulation or regulatory adjustment of the immune system. It has natural and human-induced forms. The membrane attack complex, MAC. Yeah, this was mentioned above here. So basically activation, the end result of the complement activation or complement fixation cascade. Part of the complement system is activation of the cell-killing membrane attack complex. So the membrane attack complex, MAC, or terminal complement complex, TCC, is a complex of proteins typically formed on the surface of pathogen cell membranes as a result of the activation of the host's complement system, and as such is an effector of the immune system. And here is added a note on this word effector. So an effector molecule is usually a small molecule that selectively binds to a protein and regulates its biological activity. In this manner, effector molecules act as ligands, so around a central point, that can increase or decrease enzyme activity, gene expression, or cell signaling. Effector molecules can also directly regulate the activity of some mRNA molecules. It's called ribose switches. In some cases, proteins can be considered to function as effector molecules, especially in cellular signal transduction cascades, so the complement system. The term effector is used in other fields of biology. For instance, the effector end of a neuron is the terminus or end of the neuron where an axon makes contact with a muscle or organ that it stimulates or suppresses. Very fascinating stuff here. And this is this MAC is a complex of proteins. It's a bunch of proteins formed on the surface of a pathogen cell membranes as a result of the activation of the host complement system. So the complex goes on that pathogen membrane, which is pretty interesting. Yeah, so it selectively binds to it and can alter its activity and so on. So the globulins right here, so we're mentioned over here. Yeah, scrolling up here. So the 50 protein and protein fragments that make up the complement system, they account for about 10% of the globulin fraction of blood serum. So the globulins are a family of globular or spherical proteins that have higher molecular weights than albumins and are insoluble in pure water but dissolve in dilute salt solutions. And albumin is a family of globular proteins, the most common of which are the serum albulins. So they're also spherical proteins. All the proteins of the albulin family are water soluble, moderately soluble in concentrated salt solutions, and experience heat denaturation. And a denaturation is a process in which proteins or nucleic acid lose the quaternary structure, so that higher structure, or tertiary and secondary structure, which is present in their native state, by application of some external stress or compound such as a strong acid or base. A concentrated inorganic, salt as well, or an organic solvent, e.g. alcohol or chloroform, and agitation and radiation or heat. So basically denaturation, proteins, and these lose their structure and goes back to their basic building block setup, or just not in this more advanced structure. It goes back into their just basic chains. Inflammation, from Latin, and here if we scroll up here, so this was mentioned here, part of the complement system as well, inflammation to attract additional phagocytes. So inflammation, over here from Latin, inflammatito, let's see the pronunciation here.
Inflammatio.
Inflammatio is part of the complex biological response of body tissues to harmful stimuli, such as pathogens, damaged cells, or irritants, and is a protective response involving immune cells, blood vessels, and molecular mediators. The function of inflammation is to eliminate the initial cause of cell injury, clear out necrotic cells or dying cells, and tissues damaged from the original insult and the inflammatory process, and initiate tissue repair. Yeah, so it's a complex biological response. And it's protective response involving immune cells, blood vessels, etc., a whole bunch of stuff. And it's to eliminate the initial cause of cell injury, clear out dead cells and tissues damaged from both the insult and the inflammatory process, and also initiate tissue repair. Pretty interesting stuff here. And now this irritants right here, so harmful stimuli such as pathogens, damaged cells, or irritants. Irritation in biology and physiology is a state of inflammation or painful reaction to allergy or cell lining damage. A stimulus or agent which induces the state of irritation is an irritant. Irritants are typically thought of as chemical agents, for example, phenol and capsaicin, but mechanical, thermal heat, radiative stimuli, for example, ultraviolet light or ionizing radiations, can also be irritants. Irritation also has non-clinical usages referring to bothersome physical or psychological pain or discomfort. And allergies, also known as allergic diseases, are a number of conditions caused by hypersensitivity of the immune system to typically harmless substances in the environment. Pretty interesting stuff here. All right, continue further. So ionizing radiation, or ionizing radiation with a S instead of a Z, including nuclear radiation, consists of subatomic particles or electromagnetic waves that have sufficient energy to ionize atoms or molecules by detaching electrons from them. The particles generally travel at a speed that is 99% of that of light, and the electromagnetic waves are on the high energy portion of the electromagnetic spectrum. It's pretty interesting. So it's basically a near light speed traveling at, of that of light there. And basically subatomic particles or electromagnetic waves. And necrosis is basically necrotic cells right here. Let's bold that. Necrosis from ancient Greek, this word meaning death, is a form of cell injury which results in the premature death of cells in living tissues by autolysis. Autolysis, you see this?
Autolysis.
Or autolysis. Necrosis is caused by factors external to the cell or tissue, such as infection or trauma, which result in the unregulated digestion of cell components. In contrast, apoptosis, see the pronunciation here.
Apoptosis.
Yeah, apoptosis is a naturally occurring programmed and targeted cause of cellular death. While apoptosis often provides beneficial effects to the organism, necrosis is almost always detrimental and can be fatal. So, necrosis is a premature death of cells caused by injury, external factors, infection, trauma, etc., and unregulated digestion of cell components. Which result in unregulated digestion of cell components. But apoptosis is a naturally occurring programmed and targeted cellular death. Very interesting. And then now this is by living tissues by autolysis. So autolysis, more commonly known as self-digestion, refers to the destruction of a cell through the action of its own enzymes. It may also refer to the digestion of an enzyme by another molecule of the same enzyme. Yeah, so let's see that again. So it refer to the digestion of an enzyme, so eating that enzyme or absorbing it by another molecule of the same enzyme. Yeah, so an enzyme eating itself is pretty interesting. Or also you could have this self-digestion refers to the destruction of a cell through the action of its own enzymes. Very, very interesting. So going further now we finally go through this. Is a whole bunch of MES notes here. And notice how the indent is MES notes there all the way after this complement system. And there's a phagocytosis. So anyways, let's go back here. So jawed vertebrates, including humans, have even more sophisticated defense mechanisms, including the ability to adapt to recognize pathogens more efficiently. Adaptive or acquired immunity creates an immunological memory, leading to an enhanced response to subsequent encounters with that same pathogen. This process of acquired immunity is the basis of vaccination. All right, now continue further here. Let's look at these jawed vertebrates. The term for it is a gnathostomata. See the pronunciation here?
Gnathostomatus.
Or gnathostomata, or the jawed vertebrates. The term derives from Greek gnathos or nathos. See this? Nathos. Okay, I guess they don't pronounce the G, which means jaw, and stoma meaning mouth, so mouth jaw. Gnathostome diversity comprises roughly 60,000 species, which accounts for 99% of all living vertebrates. Now let's look at this immunological memory. Immunological memory is the ability of the immune system to quickly and specifically recognize an antigen that the body has previously encountered and initiate a corresponding immune response. Generally, these are secondary, tertiary, and other subsequent immune responses to the same antigen. Immunological memory is responsible for the adaptive component of the immune system, special T and B cells, the so-called memory T and B cells, and immunological memory is the basis of vaccination. Emerging research shows support for the innate immunities, cell participation in immune memory responses in vertebrates as well as vertebrates. So basically, they're saying research or new evidence shows that the innate system also participates in the immune memory responses, so the non-innate ones. So invertebrates as well as vertebrates. Interesting. So now here is a diagram here, the time course of an immune response. Due to the formation of immunological memory, so reinfection at a later time leads to a rapid increase in antibody production and effector T cell activity or targeted attachment to proteins and other stuff. Yeah. Anyways, let's look at this. So these later infections can be mild or even unapparent, which is not apparent or inapparent. And this is involving antibody production. So you have initial response here. Here's first exposure. There's a boost in antibodies and effector T cells, which go after the pathogens. And then you have weeks over here. And then so that's the initial response is high. And then as it goes down, here's an inapparent infection, which you don't notice that you're, quote, infected. But the T cells go up. There's protective immunity. So it goes up here. So it goes up quickly. And then what happens here is, then after years, you have the memory. So it's protective immunity. Then you have memory right here. You have mild or inapparent reinfection. And then there's a spike in that antibody and effector cells count. So very, very interesting. So it supposedly memorizes them. Anyways, continue further. So white blood cells, or WBCs, and this we're going to get to this T cell and B cells. So these are all white blood cells. So memory T and B cells, we'll get to those in a bit. So, but first we need to go over white blood cells. So white blood cells, WBCs, are also called leukocytes. See the pronunciation?
Leukocytes.
Yeah, leukocytes, or here, this spelling with a C.
Or leukocyte.
Leukocytes, also called leukocytes, are the cells of the immune system, also called immune cells, that are involved in protecting the body against both infectious disease and foreign invaders. All white blood cells are produced and derived from multipotent cells in the bone marrow, and known as hematopoietic stem cells.
Hematopoietic
stem cells. Leukocytes are found throughout the body, including the blood and lymphatic system. And so they're found throughout the body, including the blood, lymph, etc. Hematopoietic stem cells, HSCs, are the stem cells that give rise to other blood cells. This process is called hematopoiesis. Hematopoiesis, all right. Yeah, so they're derived from these multipotent cells known as hematopoietic stem cells that give rise to these other ones. So they're unspecialized, and they specialize into these other blood cell types. And this multipotency describes progenitor cells which have the gene activation potential to differentiate into discrete cell types. Yeah, so it can differentiate itself into specialized cells. So bone marrow is a semi-solid tissue found within the spongy, also known as cancellous. See the pronunciation here?
Cancellous. Portion.
Let's try that again.
Cancellous.
Or cancellous. Let's call it. Okay, cancellous portion of bones. In birds and mammals, bone marrow is a primary site of new blood cell production, or hematopoiesis. See this again? Hematopoiesis. The composition of marrow is dynamic, as a mixture of cellular and non-cellular components. The connective tissue shifts with age and responds to systemic factors. In humans, marrow is colloquially or informally characterized as red or yellow, depending on the prevalence of hematopoietic cells, blood cells, versus fat cells. The more hematopoietic cells, that's called red, and the more fat is yellow there. And here's the setup here of this bone marrow. So structure of a long bone. Here's the segments here. This is the articular cartilage, this proximal epiphysis. There's metaphysis. There's a spongy bone. Epiphyseal line, this line here, and this red bone marrow right here with more hematopoietic stem cells. Then this is the endosteum, compact bone. Then you have this, this is the compact bone at the side there. Medullary cavity. And there's a yellow bone marrow, with more fat cells. And then this right here is the periosteum. This segment is called the diaphysis. There's the metaphysis distal at the far end. epiphysis, and there's articular cartilage, and there's nutrient artery. Very, very interesting setup. So anyways, the structure of a long bone. I'm not going to go over the other terms in there, but I just wanted to illustrate there's the red bone marrow and the yellow bone marrow, and then the basically the red has higher hematopoietic cells, blood cells, and then the fat cells is the yellow one, more of those. All right, let's continue further. So a lymphocyte is a type of white blood cell in the immune system of jawed vertebrates. Lymphocytes include natural killer cells which function in cell-mediated cytotoxic innate immunity, and then it says T cells for cell-mediated cytotoxic adaptive immunity, so that's T cells, natural killer cells for innate, T cells for adaptive, and B cells for humoral antibody-driven adaptive immunity, or antibody-driven, and then it's adaptive. That's the B cells, memory, T and C, memory or adaptive. They are the main type of cell found in lymph, which promoted the name lymphocyte. So there's the lymph there. Lymphocytes make up 18% and 42% of circulating white blood cells. Interesting. So white blood cells, a big portion is these lymphocytes. And cytotoxicity is the quality of being toxic to cells or damaging to cells. And then here's cell-mediated immunity. So here's the cell-mediated is T cells, and then the other one's antibody-driven or mediated. Cell-mediated immunity is an immune response. Yeah, this other one, killer cells, also cell-mediated. So cell-mediated immunity is an immune response that does not involve antibodies. Rather, cell-mediated immunity is the activation of phagocytes, antigen-specific cytotoxic T-lymphocytes, and the release of various cytokines in response to an antigen. This basically involves, the phagocytes, which is just engulfing the pathogen with cell membrane and so on, or other methods. That's a cell membrane. And then also cytokines, you have these small proteins, part of the complement system. And you also have antigen-specific cytotoxic T lymphocytes just go and attack the antigens, as opposed to the antibodies that remember a pathogen or antigen, and then it basically connects to them. And let's have a refresher on antibodies. Antibodies. Let's go all the way back. So antibodies. So an antibody AB, also known as immunoglobulin Ig, is a large Y-shaped protein that is used by the immune system to identify and neutralize foreign objects, such as pathogenic bacteria and viruses. The antibody recognizes a unique molecule of the pathogen or organism that can produce a disease, called an antigen, AG, or antibody generator. And you have that system right here. So antibody, and you have the antigen binding sites, and you connect to these antigens, and it can memorize. It gets specific to them, and it memorizes them in a lock and key theory. All right, here we just teleported back here. So now let's look at natural killer cells, and that's the cell-mediated cytotoxic innate. The key one is the innate immunity. So natural killer cells, also known as NK cells or large granular lymphocytes, LGL, are a type of cytotoxic lymphocyte critical to the innate immune system and represent 5 to 20% of all circulating lymphocytes in humans. Interesting, yes. And human natural killer cell, colorized scanning electron micrograph right here. And going over a T cell. T cell is a type of lymphocyte. T cells are one of the important white blood cells, or it should be one of the most important. One of the I'm just gonna put this here. Yes, so one of the most important white blood cells of the immune system and play a central role in the adaptive immune response. T cells can be distinguished from other lymphocytes by the presence of a T cell receptor, TCR, on their cell surface. T cells are born from hematopoietic stem cells found in the bone marrow, and developing T cells then migrate to the thymus gland to develop or mature. T cells derive their name from the thymus, so that thymus gland, after migration. This was a mistake there on my part. After migration to the thymus, the precursor cells mature into several distinct types of T cells. T cell differentiation also continues after they have left the thymus. Groups of specific differentiated T cell subtypes have a variety of important functions in controlling and shaping the immune response. Alright, so this T cell receptor is a protein complex found on the surface of T cells or T lymphocytes, that is responsible for recognizing fragments of antigen as peptides bound to major histocompatibility complex MHC molecules. And these are cell surface proteins essential for the adaptive immune system. Yeah, so they're basically these T cell receptors are found on the surface T cells, and that is responsible for recognizing fragments of antigen as peptides bound to major, or this MCH molecules, which are cell surface proteins as well. Yeah, so basically it's recognizing these fragments of antigen, peptides, bound to these other molecules right here, and there are also cell surface proteins there. The binding, so the binding between TCR and antigen peptides is of relatively low affinity or attractive force and is degenerate. That is, many TCRs recognize the same antigen peptide, and many antigen peptides recognized by the same TCR. So yes, the binding between them is relatively low affinity, attractive force, and is degenerate. Yeah, and this is referring to this degenerate. So many TCRs recognize the same antigen peptide, and many antigen peptides recognize the same TCR. Note that within biological systems, degeneracy occurs when structurally dissimilar components, modules, pathways, it can perform similar functions, i.e. are effectively interchangeable under certain conditions, but perform distinct functions in other conditions. Degeneracy is thus a relational property that requires comparing the behavior of two or more components. In particular, if degeneracy is present in a pair of components, then there will exist conditions where the pair will appear functionally redundant, but other conditions where they will appear functionally distinct. So basically, as a setup where different components can perform similar functions. So, but then they can also change. But other conditions where they'll appear functionally distinct. T cells that successfully develop react appropriately with MHC. That's at these major histocompatibility complex molecules. Successfully develop T cells that successfully develop react appropriately with MHC immune receptors of the body, called positive selection, and not against proteins of the body, called negative selection. Yeah, so that's interesting. So these ones that successfully developed, they can, so they react appropriately with these MHC and not against proteins of the other ones. But then if it's not successfully developed, you can have a different case where it's dissimilar. It's similar, it's T cell, but then it's not reacting appropriately to these MHC immune receptors of the body. So then, yes, and not against proteins. So if it does react against the proteins of the body, that's called negative selection. Positive is if it reacts with these immune receptors. Anyways, that's pretty interesting. So the thymus is a specialized primary lymphoid organ. Yeah, not the gland. I think I mentioned thymus gland, but anyways, so the thymus is a specialized primary lymphoid organ of the immune system within the thymus. Within the thymus, thymus cell lymphocytes or T cells mature. The thymus is located in the upper part of the chest and in front of the heart. The thymus is made up of immature T cells called thymocytes, as well as lining cells called epithelial cells. So lining cells of the inner organs, which help the thymocytes develop. So thymus is a primary lymphoid organ, and it has within it has thymus cell lymphocytes or T cells. So thymus cell, so T cells, and is located in the upper part of the chest. And let's see. And made of immature T cells called thymocytes, and as well as lining. Yes, thymus is made up of immature ones, and within it contains these T cells that are mature, as well as lining cells called epithelial cells, which help thymocytes develop. And it was very, very interesting. So here is the thymus in front of the chest, and you have thymatic lobule there, interlobular septum. This is basically a section there, and this is that section right there, interlobular septum. Medulla is a medulla inside. There's a cortex, and this is called a capsule, and there's a thymic corpuscle. Interesting. And here's a scanning electron micrograph of a human T cell. Yes, looks like a giant blob there. And let's continue further. And here's a size comparison. A scanning electron micrograph of a red blood cell left, a platelet center, and a T cell, a T lymphocyte on the right, colorized, just to show the contrast. All right, that's pretty amazing stuff there. So that's a platelet, for blood clots and so on. Red blood cell, and this right there. Yes, anyways. So that's T cell. This one has usually iron and stuff like that, and sends nutrients and so on. Yeah, and also deliver the oxygen to the body. So that's a red blood cell. So B cells, also known as B lymphocytes, are a type of white blood cell of the lymphocyte subtype. They function in the humoral immunity component of the adaptive immune system. B cells produce antibody molecules. However, these antibodies are not secreted. Rather, they are inserted into the plasma membrane, where they serve as part of B cell receptors. When a naive or memory B cell is activated, so naive or memory is activated by an antigen, it proliferates and differentiates into an antibody secreting effector cell known as a plasmablast or plasma cell. Additionally, B cells present antigens. They are also classified as professional antigen presenting cells, APCs, and secrete cytokines, so those small proteins. In mammals, B cells mature in the bone marrow, which is at the core of most bones. In birds, B cell mature in the Bursa of Fabricius. See the pronunciation of this whole thing.
Bursa of Fabricius.
Oh yes, Bursa of Fabricius, a lymphoid organ where they were first discovered by Chang and Glick, which is why the B stands for Bursa, not bone marrow, as commonly believed. Well, I should change it by now. Humoral immunity is the aspect of immunity that is mediated by macromolecules found in extracellular fluids, such as secreted antibodies, complement proteins, which are part of the complement system, and certain antimicrobial peptides. Yeah, so this is this type of immunity is mediated by these macromolecules found in these extracellular, so not in the cells. Yeah, and you have secreted antibodies, these proteins, and other antimicrobial peptides. There's B cells, unlike the other two classes of lymphocytes, T cells and natural killer cells, express B cell receptors, BCRs, on their cell membrane, and BCRs allow the B cell to bind to a foreign antigen against which it will initiate an antibody response. It binds to it and it can initiate antibodies to it. The B cell receptor BCR is a transmembrane protein, so it's all around the surface. On the surface of a B cell through so it's a transmembrane protein on the surface of B cell. Through biochemical signaling and by physically acquiring antigens from the immune synapses, the BCR controls the activation of the B cell. BCR controls the activation of the B cell. B cells are able to gather and grab antigens by engaging biochemical modules for receptor clustering, cell spreading, generation of pulling forces, and receptor transport, which eventually culminates in endocytosis and antigen presentation. And recall endocytosis from earlier. That's just over here. So endocytosis is the process of bringing in substances into the cell, and phagocytosis is using the cell membrane to grab it. So let's just go back here now. Let's go back. Why is it not going? It's frozen. Oh, we're back, so it's working now. And let's click here and go there. Yes. And now let's look at this antigen presentation. So this is from openmd.com. So antigen presentation is process by which an antigen is presented to lymphocytes in a form they can recognize. Antigen presenting cells may ingest or digest the antigen and present the fragments to the cell surface for recognition by the lymphocytes. Now this is from this. Now we'll go back to Wikipedia. So antigen presentation is a vital immune process that is essential for T cell immune response triggering. So because T cells only recognize only fragmented antigens displayed on cell surfaces, antigen processing must occur before the antigen is transported to the surface of the cell, and this process is known as presentation. So before it's transported, it has to process that antigen. In other words, recognize it before it turns into fragments. So before the antigen fragments are transported, where it can be recognized by a T cell receptor. If there has been an infection with viruses or bacteria, the cell will present an endogenous inside or exogenous or from the outside peptide fragment derived from the antigen by MHC molecules. That's a major histocompatibility complex molecules. B cell receptors, however, on the surface of B cells bind to intact, native, and undigested antigens, so not the fragments, of a structural nature, rather than to a linear sequence of a peptide which has been digested into small fragments and presented by the MHC molecules, such as the T cells. I'll put that here. Such as in T cells. Or more accurately, such as in the case of T cells then. Going further, so we mentioned this over here. So then this is through biochemical chemical signaling and physically acquiring antigens from the immune synapses. So now let's go over what those are. Let's bold this synapses right here. All right, synapses. An immunological synapse or immune synapse is the interface between an antigen-presenting cell or target cell and a lymphocyte such as a T/B cell or natural killer cell. Yeah, so it's similar to that electrical synapse, and it's basically that interface or that junction between the two. So the B cells, you have the lymphocyte and the antigen-presenting cell or target cell. So the interface was originally named after the neuronal synapse with which it shares the main structural pattern. An immunological synapse consists of molecules involved in T cell activation, which compose typical patterns activation clusters. Immunological synapses are the subject of much ongoing research. Interesting. So an antigen-presenting cell or APC or accessory cell is a cell that displays antigen bound by major histocompatibility complex MHC proteins on its surface. This process is known as antigen presentation. The T cells may recognize these complexes using their T cell receptors, TCRs, and APCs process antigens and present them to T cells. And this processing, so antigen processing or the cytosolic pathway, let's see the pronunciation here.
Cytosolic pathway.
Yes, so cytosolic pathway is an immunological process that prepares antigens for presentation to special cells of the immune system called T-lymphocytes. It is considered to be a stage of antigen presentation pathways. That's very interesting. Continue further here. Here is a transmission electron micrograph of a human B cell. So yes, this is I believe a cutout, and you can see the insides of it, and so on. That's pretty interesting stuff, and that's 500 nanometers there. So it's pretty big, yeah, relative to the other viruses, etc. So this is quite big there. I believe it's a cross section there. That's what I would assume. Anyways, in bird, yeah, we're going over that Bursa of Fabricius. Let's see this pronunciation again.
Fabricius.
Yeah, the Bursa of Fabricius. And remember, this one is where the B comes from for B cells, not actually bone marrow. Although I should change it. Let's go back over here. Yeah, so we're back here. So in birds, B cells mature in the Bursa of Fabricius. And that's a lymphoid organ where they were first discovered by Chang and Glick, which is why the B stands for Bursa and not bone marrow. All right, as commonly believed. So let's go over that Bursa right here. In birds, the Bursa of Fabricius, Latin Bursa. This is right here. Bursa is this word cloacalis.
Cloacalis or
Cloacalis or Bursa.
Bursa Fabricii.
Or Bursa Fabricii is the site of hematopoiesis. Let's see the pronunciation again.
Hematopoiesis.
Hematopoiesis is a specialized organ that, as first demonstrated by Bruce Gulick and later by Max Dale Cooper, and Robert Good, is necessary for B cell part of the immune system development in birds. Mammals generally do not have an equivalent organ. The bone marrow is often the site of bone hematopoiesis and B cell development. Hence why it's commonly called bone marrow, even though it came from the bursa, right there, the B cells. And B cell development in the bone marrow. The bursa is presented in the cloaca of birds and is named after Hieronymus Fabricius. See the pronunciation here.
Hieronymus Fabricius.
Yeah, Hieronymus Fabricius. This is May 20th, 1533, and he died in May 21, 1619. Who described it, and lecture notes found and published in 1621. Or here's at the end. So who described it in lecture notes found and published in 1621. So after his death, they were found. But yeah, so he died 1619. But the lecture notes were found here, and it talked about the Bursa of Fabricius. So anyways, yeah, I mean, he talked about the Bursa, yeah, of birds were the site of B cells, B cell growth and development. So anyways, a cloacia,
Cloaca.
or cloaca, or plural cloacae.
Cloacae.
The cloacae is a posterior or backside orifice or opening that serves as the only opening for the digestive, reproductive, and urinary tracts, if present, of many vertebrate animals. So that's that external opening, and that's where it's found on there. So the bursa is present in the cloaca of birds. So basically, this opening right there. The Bursa of Fabricius is the source of B cells in chickens. There's a thymus there for T cells, and now you have this right here, the Bursa of Fabricius, right at next to that cloaca, or that basically the opening to the digestive system, and it's also one opening for the digestive, reproductive, and urinary tracts, if present of many vertebrate animals. Pretty interesting. This is from FortisLife.com. Very, very interesting. Let's continue further, and now here's a breakdown of all the types of the white blood cells and so on. So here's a diagram showing the development of different blood cells from hematopoietic stem cells to mature cells. So we have this right here is the multipotential hematopoietic stem cell, also called a hemiocytoblast. Then it can split up into a common myeloid progenitor cell, that can generate further down the line here. And also you have the common lymphoid progenitor cell. So there's myeloid. This gets these red blood cells there. And here, let's just do a zoom in and scroll. All right, so zoom in and scroll. And so this is common lymphoid progenitor. Let's look at this side. So you have the natural killer cell, or large granular lymphocyte. And then you have the small lymphocyte right here. And in these small lymphocytes, you can form a T lymphocyte or B lymphocyte. And from there, you get a plasma cell for the B lymphocyte there. And so now if you go back to this common myeloid progenitor cell, you get these red blood cells there. So this is the erythrocyte. So the red blood cells are over there. And you also can get a mast cell, a myoblast, or a myeloblast. Then this can form a basophil or neutrophil, an eosinophil, and monocyte. And there's a macrophage. And then over here is a macrokaryocyte. And there's a thrombocytes over there. So yes, you can get this supergiant one. It goes over here. So this is a diagram, showing the development of different blood cells from hematopoietic stem cells to mature cells. Going further, we discussed a native B cell here, a naive B cell. Let's go back to that naive B cell. It was somewhere over here. Yeah, so we described here. So when a naive or memory, versus memory, is activated by an antigen, it proliferates and differentiates into an antibody secreting effector cell known as a plasmablast or plasma cell. So there's a plasma cell there. And that is when it gets activated by antigen, proliferates and differentiates into that. And so going back over here, so once it gets activated, you get this B cell going to this plasma cell. And then a naive B cell is a B cell that has not been exposed to an antigen. Once exposed to an antigen, the naive cell either becomes a memory B cell to keep in memory for a later time, or a plasma cell that secretes antibodies specific to the antigen that was originally bound. Plasma cells do not last long in the circulation. This is in contrast to memory cells that last for very long periods of time, and memory cells do not secrete antibody until activated by their specific antigen. Very interesting. So basically, these memory cells circulate around for a long time, and for long periods of time, and then once activated by the antigen, it gets presented again. It turns to the plasma cell, and then fires off antibodies at it, and doesn't last too long. That's very, very interesting. So a memory B cell, or MBC, is a type of B lymphocyte that forms part of the adaptive immune system. Memory B cells circulate in the bloodstream in a quiescent, or inactive, let's see the pronunciation.
Quiescent.
Yeah, quiescent or inactive state, sometimes for decades. Oh, yeah, bold up. Their function is to memorize the characteristics of the antigen that activated their parent B cell during initial infection, such that if the memory B cell later encounters the same antigen, it triggers an accelerated and robust secondary immune response. Memory B cells have B cell receptors, BCRs, on their cell membrane, identical to the one of their parent cell, that allow them, it's just a regular B cell, that allow them to recognize antigen and mount a specific antibody response. Differentiation of memory B cells into plasma cells is far faster than differentiation by naive cells. So yes, that's interesting. So the memory ones can go super quickly to the plasma cells, but this one is slower, which allows memory B cells to produce a more efficient secondary immune response. Very, very interesting setup here. So let's make this smaller and take a look at this figure. Yeah, so we have B lymphocytes are the cells of the immune system that make antibodies to invade pathogens like viruses. Yes, they make the antibodies there and then attack to the antigen generators or the pathogens. So they form memory cells that remember the same pathogen for faster antibody production in future infections. So here's a naive cell right here, and here's memory B cells specific for virus A. All right, so what this figure is saying, I think what it's basically saying is basically the naive cell that gets exposed to this right here. We call this virus A, but it says A-1 there. I couldn't find why it says that in this diagram. But anyways, and then what happens is this can turn into a memory cell where it remembers, this virus right there. So then it can create, when it gets exposed to a new one, or later on of the same virus or virus type, and then it can produce antivirus A antibodies, the exact ones it can produce, and then attack this stuff right there. So yes, this diagram is a bit simple and a bit just not detailed enough. Yeah, but anyways, let's continue further. So now let's look at memory T cells. They are a subset of T lymphocytes that might have some of the same functions as memory B cells. So this might interesting part. Their lineage is unclear. Primary function of memory cells is augmented immune response after reactivation of those cells by reintroduction of relevant pathogen into the body. So here is a diagram, processes in the primary immune response. So let's take a look over here. Here's the whole setup of these processes right here. Let's zoom in. So what we have is macrophage adopts the pathogen. This is phagocytosis. So it's that it's a large white blood cell. We'll get to that in a bit. Basically, so then phagocytosis grabs this pathogen, i.e. a virus, quote, antigen, which generates antibodies. So it engulfs it. Then pathogen is broken down into components in the phagolysosome. So put that inside there. And then what happens is macrophage presents components of the pathogen to a T lymphocyte, and then it presents it to it. There's a receptor protein on this T lymphocyte, and presents components of it, so those peptide fragments or protein fragments to it. And then what happens is you have activation of either a killer cell, cytotoxic T cell destroys the infected body cell. So it goes in this part right here, goes in and destroys them. Yeah, and then so it presents it to this, and now it goes and destroys whatever cells that are infected by this pathogen. So killer cell goes and kills all these, and yes, and this just dies out over there. And then what happens? Here's macrophages phagocytize and digest. So then this goes and eats this over here and breaks that apart. And then it could also go to a B lymphocyte. So that was the T lymphocyte. So the T lymphocyte could activate the B lymphocyte. So first, specific antibodies against the antigen. So those ones get presented over to this B lymphocyte. And then what happens is this can either proliferate into plasma cells, and then they produce specific antibodies to stop these viruses or antigens right there. And then antigen antibody complexes right there. So antigen antibodies, and then the macrophage again phagocytizes and digests this broken up stuff right there. But this B lymphocyte could also, instead of going to a plasma cell, this is a naive one, so this can go straight to a memory one. And then memory cells can later induce the secondary immune response upon renewed contact with the same pathogen. Yeah, so same pathogen. This one, so then the basic B cells kill the antigen, then killer cells kill the cells infected by the antigen. Very, very interesting. And yes, processes in the primary immune response. All right, let's continue further. So MES note macrophages, these ones right here is pretty much primary role. Abbreviated M and then this Greek letter there. Yeah, this Greek letters hover over it. That is phi. Let's see this. Yeah, phi. So Greek M phi or M capital phi there, or MP for phi is P, PHI. So Greek large eaters from Greek macros equals large, and phagogen. See the pronunciation here.
Phagine.
Or vagine means to eat. So large to eat. Large eaters are a type of white blood cell of the immune system that engulfs and digests anything that does not have on its surface proteins that are specific to healthy body cells, including cancer cells, microbes, cellular debris, foreign substances, etc. The process is called phagocytosis, which acts to defend the host against infection and injury. So there's these the macrophages. So macrophage stretching its arms, called the philopodia. Is this pronunciation?
Philopodia.
Or philopodia, to engulf two particles, possibly pathogens, in a mouse. So there's this right here, the macrophage, and extends its arms and eats a bunch of small particles. A very, very interesting setup there.